Okay, welcome to the online series for biology grade 12, the ICB online program for YouTube classes. This is Muhammad Hassan Shafiq, and I welcome you to the lecture 11 of Unit 23 labeled The Chromosome and DNA. Today, in this lecture, we will be covering the DNA as the hereditary material and the Meselson and Stahl experiment for DNA replication. Moving on to today's slide, we will start our chapter with DNA as the hereditary material. Now, initially, it was thought that maybe proteins have something to do with the transmission of characters or the traits which we inherit from our ancestors are due to uh, the proteins which we inherit. And when the chromosomal theory of inheritance was put forward in 1910 and confirmed, the geneticists started thinking over the issue uh, of the form hereditary units and found in chromosomes. Now we do know that chromosomes are composed of both DNA and protein. On which of these was a hereditary information written? So, as you know, the proteins associated with the chromosomes are histones, and we have DNA as well. So, over a period of 30 years, starting in the late 1920s, a series of investigators actually addressed this issue, which was very important at the time, as there was no much clarity regarding what actually is the substance which determines your hereditary information. So this experiment, which was put forward in 1928 by Frederick Griffith, is very important from this aspect that it first, actually, for the first time, gave an idea that DNA has something to do with the hereditary information and not protein. And he made some unexpected observations while studying a bacterium, Streptococcus pneumoniae, which exists in two types. Now, this bacterium causes pneumonia in mice and usually exists as two forms. The first type, the S type, has a polysaccharide capsule and it forms a smooth and shiny colony on a culture medium, hence the name S type. The other form, the non-virulent, non-pathogenic form, which lacks the information regarding the polysaccharide capsule synthesis, it was the R type. Now, Griffith carried a series of experiments. In the first one, he injected healthy mice with a strain of S type, and expectedly, the mice died. But when he injected similar mice with the R type strain, the mice showed no ill effects. So, on the basis of these observations, it was clearly evident that the S type might be associated with the death, as it had the capsule. So the capsule probably had something to do with virulence. Uh, as a control, he on the second in a second experiment, he injected another group of mice with the R type, the other form, and the mice showed no ill effects and remained perfectly healthy. So, as a control, Griffith injected heat killed S type, in that he destroyed all the polysaccharide capsules and all the proteins. Obviously, they must have been destroyed by the heat. He actually wanted to see if the polysaccharide capsule itself had a virulent effect. The mice remained perfectly healthy after the injection of the heat killed S type, which confirmed that maybe the polysaccharide capsule had something to do with the death of the mice. As a final control, he blended living R type with heat killed S type, okay, both of which initially did not harm the mice. And to his surprise, when he injected heat killed S type with the live R type, the mice died. And the blood of these mice were found to contain a large number of S type virulent bacteria, which had surface protein characteristics of the living previously R type strain. So, uh, this was actually quite shocking at that time, that actually what resulted in the death of the mice, and where exactly did this live S type come from when it was not injected to the mice in the first place at all. So, on the basis of these unexpected observations, Griffith concluded that somehow there was something which was transferred or passed out from the heat killed S type to the live R type. And this substance, whatever got transferred, converted that live R type into an S type form. So, this transfer of genetic material from one organism to another, by which genetic makeup of the recipient is altered, is called transformation. Now, I'll very quickly go through figure 23.4, which gives you an overview of this experiment. In the control, he injected living S-type bacteria into the mice. They died, and their blood was composed to have this virulent S-type form. In another experiment, he injected the living R-type, and the mice remained perfectly fine. In a third experiment, as a control, he injected heat killed S type into the mice, and the mice remained perfectly healthy. But in the fourth experiment, he tried to mix the second and third experiment in a way that he injected mice with a live R type and a heat killed S type into the mice. A substance from the heat killed must have been transformed, converting the R type into the virulent S type, which resulted in the death of the organism. This was a very good indication that proteins might not have been the case here, because they must have been destroyed uh, during the heat treatment. But still, there was a lot to be proved, and this was done later during the Hershey Chase experiment. So they did a very convincing experiment on bacteriophages in the year 1952 by the names I mentioned, Alfred Hershey and Martha Chase. Now, they worked on, like I said, bacteriophages. These are the viruses that attack bacteria, like the name indicates, bacteria eater. Their body consists of DNA and protein. Now, during infection, they multiply in the host and make many copies within like 20 to 25 minutes. It was not known until 1952 that whether DNA or RNA, which possesses the hereditary information of bacteriophages. Even scientists were not sure. During infection, the whole viral particle usually remains outside the body, and only the DNA or protein gets entry. So we do know now that it's obviously the viral or phage DNA, but till that time it was not initially quite clear. In 1952, Hershey and Chase out at an experiment for this purpose. So when a bacterium gets infected by a phage, number one, whether is it the DNA or protein which gets entry, and number two is that whether is it the whole viral particle that gains access, or is it only the DNA. So this experiment was carried out in 1952, and the initial basis of this experiment was to confirm that how, uh, or actually what actually transfers from one to another, to actually, or which actually determines the hereditary information. So they actually carried out an experiment in which they labeled the DNA of bacteriophages with radioactive isotopes of phosphorus P32, and also labeled their protein codes with the radioactive isotope of sulfur S35. The labeled viruses were then allowed to infect bacteria. Soon after, the bacterial cells were separated from the media contents with the help of a centrifuge technique. Now, the media contents and the bacterial cells were analyzed for the activity of P32 and S35. In this analysis, P32 was found in the bacterial cells, while S35 was still found in the medium. These observations clearly showed that during infection, P32 DNA, labeled DNA of bacteriophage, got injected into the bacterial cell, while its S35 labeled protein code still remained outside. So, since sulfur was labeled in the protein code, it was not found in the cell, which means that it was only the DNA which gained entry into the bacterial cell, and the other contents of the viral particle remained outside. Subsequently, many viral particles released outside the host. Based on these observations, Hershey and Chase claimed that the viral's DNA, not the virus protein, was responsible for detecting the directing the production of new viruses. So this also gives you an indication that whether when a particle multiplies inside a host, it is only the DNA which specifies the information for carrying out the synthesis of new viral particles, viral proteins, not the protein code, because it remained outside the cell, and it had nothing to do with the specification or multiplying of viral particles inside the bacterium. This is an uh, overview of what actually was done. So batch one is a diagram which shows the S35 which was labeled, and in batch two it was the DNA which was labeled. The DNA was labeled with P32, and the sulfur or protein code was labeled with S35. Both these phages were then allowed to infect bacteria and grow inside a medium. When they multiplied up till a good number, they were then centrifuged to separate the contents of bacteria from the medium ones. And when the bacteria were analyzed, it was initially observed that the empty protein shells just remained outside, and since the sulfur was labeled with the protein shells, no sulfur or detection was seen within the bacterial cell, but P32 was definitely seen within the bacterium, because those were the molecules which got incorporated into the bacterial cell and multiplied in number, uh, resulting in death of the host in that case. So the information specifying or being transferred to the next generation in this case was definitely proved to be the DNA and not protein. So this gave another strong convincing evidence that it is actually the DNA which is the hereditary information which specifies all the activities of the cell present in the nucleus now as we know, and not protein. Now, the second topic, which is very important in today's lecture, and it is usually regarded as a very, very important topic from the federal board point of view, the DNA replication. Replica means to make an exactly new copy of the one initially present. So, the process of this cell synthesis of DNA molecule is called DNA replication. The DNA quantity gets doubled, or a new molecule gets synthesized during the S phase of interphase during the life cycle of a cell. The molecule of a DNA which is replicated is called the parent DNA, while the molecules which are produced during this process is called the daughter DNA. A parent DNA molecule after replication gives rise to two daughter DNA molecules. Now, there were three models which were proposed at the time. Scientists tried to find the exact answer of the discovery of DNA structure, which only after the structure, which was later proved by the Watson and Crick structure, which explained the model of DNA in 1953. Before that, uh, there were still scientists who actually working over the structure, and three models at the time were proposed: the semi-conservative, the conservative, and the dispersive model for DNA replication. Now, in the semi-conservative model, according to this model, which says that the parental DNA separates, okay, and each strand acts as a template for the synthesis of a new daughter strand. This result is DNA duplexes, both of which contain one half of the parental and one half of the daughter DNA. So we can say that 50% of the sequence is conserved, okay, and 50% is the one which is newly synthesized, hence the name semi-conservative. The second model is the conservative model, which states that the parental DNA remains intact and generates copies consisting of entirely new DNA molecules. Now, this was proposed, but now we do know that this quite doesn't quite make sense, as how can a molecule replicate, making its own copies while remaining intact? The third model at the time was the dispersive model, which says that the parental DNA molecule gets dispersed, and each of these nucleotides then come together with new nucleotides to form DNA molecules or DNA duplexes, which was also rejected. And the experiment which later confirmed that the DNA replication is actually semi-conservative was the Michelson and Stahl experiment. Now we have three diagrams in figure 23.6. As you can see, the semi-conservative, the conservative, and the dispersive model. According to the semi-conservative model, like you can see in the pink one, the red one, it half of it remains conserved, and half of the blue one, is newly synthesized. In the conservative model, the parental DNA remains intact, like the red one is like that, and it's making entirely new DNA molecules, the blue ones. The dispersive model is the third one, in which you can see that the daughter duplexes, the daughter DNA strands are actually hybrids of or mixtures of a few nucleotides from the blue, the parental one, and a few from the pink one, uh, the pink one, the parental one, and blue one, the daughter one. So that actually doesn't actually quite make sense that how exactly can this happen. Michelson and Stahl actually proved semi-conservative replication by using a heavy isotope of nitrogen N15, which became incorporated into the bacterial DNA. After several generations, the bacteria were shifted to three phases from the N15 to the N14 medium, and this actually proved that DNA replication was semi-conservative. How exactly did they prove this? They actually grew bacteria into N15 and then isolated the DNA after successive generations. The DNA at that time must was containing the nitrogen of heavy isotope N15, which when they isolated and centrifuged in a cesium chloride gradient, the N15 being denser than the N14 migrated further towards the bottom, where its density exactly matched the density of cesium. So cesium chloride is a gradient in which the cesium ions being denser migrated towards the bottom, cesium chloride half dense, half less dense on the somewhere in the middle, and the chloride ions being the lightest on the top. So they actually made a gradient to see exactly where the nitrogen matches the density of the ions there. So the N15 matched to that of the cesium ion. When they then transferred the bacteria into an N14 medium, half of it was that of the N15 and half of it was that of the N14, where where you can see in generation one that the DNA from all the bacterium was then a kind of a hybrid, like above the N15 and below the N14 region. When these bacteria were then transferred for the second time into an N14, it was seen that the half of it was still the N15 and 14, and the other half was the N14. So it is actually, since the bacterium we were growing were in N14, so we can see that from these that half of it was still that of the parent, and half of it depends upon the new isotope or the new nitrogen from where they are obtaining their nitrogen requirements. Like in this case, since we are transferring it from the N15 initially to the N14, it was initially the hybrid, and that hybrid was then taken to the N15, N14 hybrid, and N14. So this actually proved it in this way that the DNA replication is actually semi-conservative. So like the half of it remains intact, and the half of the duplex then remains is the one which is newly synthesized. And since we're growing nitrogen in N14, that is why the second strand or the second part of the duplex in the newly synthesized strand comes from that of the source in which the nitrogen is present. We can divide also this experiment in 0, 20, and 40 minutes. At the 0 time, it was N15. At the 20 time, then it was N15 and 14. And at the 40th minute, it was N15 and 14 one and N14 the other. So one of the strands was a hybrid and the other was N14. So on the basis of these above mentioned results, they claimed that the DNA replication is actually semi-conservative. Now we will continue with the DNA replication in the next slide. That was all for this lecture, and we will soon join you in the next one.